Good morning, dear students. The next topic is the Rickettsiae. The bacteria grouped as Mycoplasma, Rickettsiae, and Chlamydia are considered as the smallest of all living things. Among the three, all of them are capable of infecting humans, causing various diseases, which are quite often chronic, since they are intracellular bacteria. They cannot be grown in nutrient media but can be grown only in tissue or embryo cultures. In previous class, we learned about mycoplasma. The next one is the Rickettsiae. General characteristics. This is a small, obligate, intracellular cocobacilli. They are gram-negative bacteria, better stained with Giemsa stain. They have a cell wall bigger than virus but smaller than bacteria. They have DNA and some may have RNA. Have an ATP transport system that allows them to use host ATP. The major reservoir of this type of bacteria are arthropod vectors. Example, ticks, mites, lice or fleas. They are sensitive to antibiotics. The naming of the species. The genus Rickettsiae is named after Howard Taylor Ricketts, who studied Rocky Mountain spotter fever in the Beetroot Valley of Montana. Ricketts was devoted to his research and on several occasions injected himself with a pathogen to study their effects. Unfortunately, days after isolating the organism that he believed caused typhus, he himself died of the disease. For his remembrance, these organisms are named Rickettsiae. There are different genus belonging to the group Rickettsiae. These are Rickettsiae, Coxilia, Orientia, Ehrlichia, Bortonella. And some species belonging to this genus are Rickettsiae provazekii. These caused epidemic typhus, and Rickettsiae typhi cause endemic typhus, and Rickettsiae rickettsii causing spotted fever, and Rochalimia Quintana trench fever, Coxilia burnetii Q fever. The structure of this Rickettsiae. This is similar with the gram-negative bacteria. The cell wall is also made up of outer membrane and presence of peptidoglycan and also contain lipopolysaccharide as the microcapsule and polysaccharide coverings. These are rod-shaped, cocoid or pleomorphic with no flagella. Pleomorphic means these are structurally or basically having the features of gram-negative bacteria. And the Rickettsiae. This is a small gram-negative bacilli, obligate intracellular pathogens. The major reservoir or vectors of this bacteria are lice, fleas, ticks, mites. These bacteria are colonizers in their gut. Invertebrates colonizes vascular endothelium and reticuloendothelial system. This bacteria in vertebrates colonize ചെയ്യുന്നത് vascular endothelium reticuloendothelial system. There are mainly 5 genera in this class causing human diseases. They are Rickettsiae, Bartonella, Coxilia, Ehrlichia, Orientia. And the Coxilia do not cause skin rashes, but some others are causing skin rashes and itching. These are the major human disease causing genera: Rickettsiae, Orientia, Ehrlichia. And Rickettsiae infecting, these are a figure micro photograph showing the Rickettsiae infecting endothelial cells. The mode of transmission or replication is the in a normal epithelial cell, Rickettsiae infection of an endothelial cell. First of all, a Rickettsiae is adhered to the endothelial cell. Automatically, phagocytosis is induced. So, phagocytosis has been occurred. The bacteria entering the inside the cell. This bacteria escape from the phagosome. We are already known that phagocytosis is the process of engulfing the pathogen and lyses the pathogen by certain lytic enzyme. In the case of Rickettsiae, these Rickettsiae bacteria are escape from this lytic enzyme and they can budding and colonizes. After colony formation, this bacteria can lyses the cell wall or rupturing the cell wall or killing the cell and released out. This picture showing the Phylopodium focal lysis is the another type of cell lysis, or budding is another type, and the lysis of cell of Rickettsiae provasici. And this type of infection or rickettsial infection, the symptoms are fever, headache, malaise, prostration, skin rash and hepatosplenomegaly. And these infections or rickettsial symptoms are classified typhus group, epidemic typhus, murine typhus and scrub typhus. The next one is the spotted fever group, RMSF, pox rickettsia, etc., Q fever, trench fever, ehrlichiosis. These are the some rickettsial infections. This is the example of a spotted fever group. This identified in 1906, Rickettsiae of this group multiplies in nucleus and cytoplasm. This spotted fever is transmitted by ticks. The major vector is the ticks. The spotted fever group species entering into the endothelial cells and they have multiplies in the nucleus and in the cytoplasm. So the symptoms or infections are seen like this. This is the Rocky Mountain spotted fever. The skin rashes, patches are seen on the infected area or the infected person. This is the rickettsial species and its diseases. These are the different types of rickettsial species identified and their diseases and the major reservoir or vectors of the species. The pathogenesis is clearly depicted that the infected adult tick is the vector of this bacteria. In a small wild rodents, this infected from their body released the infected adult tick. They lay eggs. Also the eggs are infected and the infected larvae biting the small wild rodents for their blood meal and the infected nymph also biting the small wild rodents for their blood meal. So these easily transmitted or bites or infected by infected humans easily. So humans are the incidental host of this infection, this bacteria. So it can easily infected by Rocky Mountain spotted fever. Life cycle of Rickettsiae is the first one is the these rickettsial colonies are released from an infected cell, and also this colony can enter into the next cell. So these cells are engulfed or induced phagocytosis. Unfortunately, these rickettsial bacteria are escape from the phagosome and they can multiply inside the cell, and after they lysis and released out the new colony. This is the life cycle of Rickettsiae. The major treatment is the chloramphenicol, tetracycline, the oxycycline, and the response within 48 hours usually. So these antibiotics can easily used for the treatment of this bacteria. And in the case of vaccine, it's developed after World War II but not commercially available. Thank you.